My name is Dr. Nicholas Boaz. I teach at North Central College in the Department of Chemistry. And today what I would like to talk to you about is the chemistry of new antiviral drugs that are used in combating the COVID-19 pandemic. While ultimately this talk is about antiviral drugs and how they can combat COVID-19, ultimately we need a little bit of background to describe how these antiviral drugs wind up working. So in this talk, we are going to discuss the cause of COVID-19 and how viruses such as SARS-CoV-2, which causes COVID-19, differ from other forms of life. In order to understand then how SARS-CoV-2 can replicate itself, we're going to talk about how information, specifically genetic information, is transferred in biological processes. We're then going to talk about how specifically the SARS-CoV-2 virus, again, which causes COVID-19, hijacks cellular machinery in order to reproduce itself. From here, we're going to discuss the drug Remdesivir, which was originally designed for a different purpose, but has been given another chance to interrupt viral replication. We're going to talk about how this drug winds up working on a molecular basis, and then finally, we're going to discuss a drug that's further up in the pipeline, but is very promising. So COVID-19 and the pandemic that it has spawned really has changed the way that American society thinks about drugs. So what I mean by this is that this new pandemic is actually causing a public health emergency that many of us in the United States have not been directly affected by in our lifetime. And so part of the reason for this is that modern medicine has really effectively limited the impact of diseases that at one point in human history were endemic in our population and have devastated human population, and it has made these diseases manageable and in many cases things of the past. So for example, the Salk vaccine has made it such that nowadays children don't have to worry about contracting polio when they're young and worried about the crippling and debilitating side effects of the polio virus. Drugs and other antiretrovirals such as Indinavir, which is also known as Crixivan, have turned HIV into something that was originally a death sentence into a disease which is now manageable. And then finally, broad-spectrum antibiotics, so for example, chloramphenicol, have made diseases which in the past were terrifying, things like the Black Plague, cholera, and typhoid fever, very rare for someone in the developed world to contract. As a side note, the reason I chose chloramphenicol over all of the other broad-spectrum antibiotics is that a North Central College alumnus was one of the first people to synthesize this drug. Her name was Mildred Rebstock, and she got her degree here at North Central College in biology. That being said, the novel coronavirus, or COVID-19, presents a challenge, specifically a virus to which we have no immunity, that we as American society haven't had to face since the HIV crisis came on the scene in the 1980s. And so, while this originally seemed like a pandemic that was limited to certain areas such as the Far East, now we can see that this disease, COVID-19, is spreading like wildfire across the United States. And so the picture I have here on the right is a picture of Illinois by county showing the incidence of COVID-19 as of mid-July. And you can see that based on the shading, so the darker the shading, the greater the number of cases, that this is present all throughout the state. Okay, so COVID-19, which is the disease that humans get, which we have termed coronavirus, is actually caused by a specific type of virus that we call SARS-CoV-2. We call it CoV-2 SARS because it's actually related to the severe acute respiratory syndrome pandemic that occurred in the early 2000s. So this is called a coronavirus because the actual virion, so when you actually take a very, very small photograph using an electron microscope of the virus, you can see, and I'll highlight this with my cursor here, that the outside of this virus sort of looks like it has a fuzz around it. So scientists originally thought that this fuzz looked like the corona of the sun during a solar eclipse, and so they termed these coronaviruses. This fuzz is actually caused by something called a glycoprotein, which is a protein with a sugar attached. That's on the outside of the virus. On the inside of the virus, we have an envelope which contains the genetic information of the virus, which the virus uses to copy itself within host cells. So the information that a virus carries is encoded, excuse me for that, is encoded in something called a nucleic acid. And in this case, the nucleic acid is termed RNA. So there are other viruses out there that are caused by RNA-containing virons, and these include things like polio, rabies, and Ebola. Very serious infections indeed. And it turns out that there are other coronaviruses as well that affect humans. Most oftentimes when a human contracts a coronavirus, they only get very mild cold-like infections. And so oftentimes the colds that you've got in the fall or the winter during cold season could indeed be caused by a coronavirus, which so in that case, they tend to be very mild. On the other hand, you can have very severe infections from things like severe acute respiratory syndrome, or SARS, or Middle Eastern respiratory syndrome, MERS. And this is now included in COVID-19 in other types of human coronaviruses. So viruses, however, work very differently than other forms of life. And so what I mean by this is that human beings, animals, plants, fungi, even bacteria are considered to be forms of life that are alive. And in these cases, the cells of these forms of life have all of the machinery required to reproduce programmed within inside themselves. So what I've shown in the top figure here is an animal cell that is going through the different stages of mitosis, which is simply the splitting of a parent cell into two daughter cells. And this is something that pretty much all forms of life that we think of as actually alive is able to do in some shape or form. It's going to look a little bit different depending upon what form of life it actually is, but it has this ability to reproduce or replicate itself using its own machinery. Viruses, on the other hand, don't have the necessary cellular machinery to replicate. And because of this, we don't actually think of viruses as actually alive. And so when you hear TV commercials saying that this certain disinfectant kills COVID-19, not really. Simply for the fact that COVID-19 as a species is not actually a form of life. It doesn't contain all of the requirements for something to be alive. And so because viruses don't contain the cellular machinery needed to replicate themselves, they have to borrow someone else's cellular machinery. And in the case of something like COVID-19, it is going to borrow the cellular machinery of your cells. It's going to enter your cells. It's going to take all of your cell building block factories that are present within your cells, and it's going to have them, instead of churning out things that keep the cell healthy, it's going to churn out copies and copies and copies and copies of the SARS-CoV-2 virus that then proliferates to other cells or to other hosts. So, in order for us to learn how viruses replicate themselves, and by extension, how scientists such as us can interrupt the cycle of replication, we need to learn more about how life encodes and uses genetic information. So how do your cells transmit information? What code makes you, well, you? So life contains its information in the form of a molecule called a nucleic acid. So if we take a look at this cell on the left here, so this cell contains at its center an information hub right here that is referred to as a nucleus. So this information hub or nucleus contains our genetic information, which is organized as a structure called a chromosome. So you can see within the cell we've got a nucleus. Within the nucleus we've got a structure called a chromosome. Chromosomes are made up of a molecule called DNA, which I've shown right here. This DNA, or deoxyribonucleic acid, is a polymer or long chain molecule that is made up of 4 smaller independent molecules, which we call monomers, and are also known as nucleotides. The order of these nucleotides, and so there are 4 of them, guanine, usually abbreviated as G, thymine, abbreviated as T, cytosine, abbreviated as C, and adenine, abbreviated as A. This order determines what information is encoded in our DNA and what makes us as a species who we are. DNA, as I show in the center here, is actually double-stranded, and it turns out this is actually very important to its function. And vital to this function is that adenine, or A, will always pair with T, or thymine. Cytosine, on the other hand, or C, will always pair with G, guanine. This is going to be vital to how the cell actually reproduces the DNA so that each of its daughter cells, when it splits, can have a full copy of its genome. So we mentioned this in the previous slide, but cells can copy their DNA. And so when a parent cell is dividing so that it turns into 2 what we call daughter cells, it's important that it's able to provide both of these daughter cells complete sets of instructions to life, which we call a genome for both cells. It does this by actually copying its complete genome or genetic information that's made of DNA. So the way in which this works is that we have an enzyme that's made out of something called protein called helicase. So this guy right here. This helicase unzips the double-stranded DNA molecule so that you have 2 single strands. Other enzymes, which are shown here and here, are called DNA polymerase. DNA polymerase actually fits new DNA building blocks that are called nucleotides. So these are these adenine, thymine, cytosine, and guanine we talked about in the previous slide. They fit these to the new strands so that you have 2 new copies. The reason that this winds up working is because the adenine on one strand, that's a single strand here, only really wants to bind to a thymine when its new strand is made. So therefore, if you have an adenine on the unzipped single strand of DNA, the DNA polymerase, our enzyme right here, knows that it has to pair a thymine or T with it. So the enzymes, which we've called in this slide helicase and DNA polymerase, are part of what we were referring to as cellular machinery. So the cellular mechanisms by which the cell is able to copy itself. And these type of mechanisms are things that viruses, generally speaking, don't have. So DNA within your cell is very similar to a central file vault or a hard drive on your computer. It contains precious material, and those of you who have ever had a hard drive die know that, well, it's not great. And so the cell needs to protect this material because without it, it doesn't have the instructions to be able to be alive. So just like you don't rip out your hard drive of your computer to share information with a colleague, you don't rip the DNA out of the nucleus to share information about the cell. Instead, the cell copies certain sections of information from its DNA genome to another nucleic acid called messenger RNA, or mRNA. This stands for messenger ribonucleic acid. This mRNA then delivers its message to other places in the cells. So mRNA, however, unlike DNA, only has a single strand. So it's going to be quite a bit different than our DNA central repository of information. So you can see in this picture here, an enzyme called RNA polymerase is able to split the DNA into 2 strands and copy a single of those strands into its mRNA form. This is called transcription. So if we're going back to the computer analogy that we started this slide with, this is very akin to copying information from your hard drive, where it's stored permanently, to your RAM, or random access memory, so that you can use it, send emails, do things like that. Okay, so we've talked about copying DNA into mRNA. Well, why would you need to go ahead and send this information around the cell? It turns out that cellular machinery is able to use this information in DNA to create building blocks that the cell needs. And one of these specific building blocks that the cell needs is called a protein. So proteins are cellular building blocks that are used in a variety of different places. They can be used to give cells structure. They can be used in signaling. They can be used to make antibodies. So we've been talking a lot about antibodies in the popular press as a way of fighting the coronavirus. It turns out that these are proteins as well. Proteins are also involved in a series of chemical reactions that convert food into chemical energy called metabolism. So the ability to make specific proteins winds up being a very vital function of the cell. And the way that the cell does this is, first of all, it takes your DNA information, like we talked about in the previous slide, and it copies this information from double-stranded DNA to a single-stranded messenger RNA using a piece of cellular machinery called an enzyme known as RNA polymerase. This messenger RNA then is able to be translated or converted into a peptide, which is just a part of a protein, using another piece of cellular machinery called a ribosome. And so from DNA to RNA, from RNA to protein, cells are able to translate what ultimately is stored genetic information into specific building blocks that the cell can use in order to live. And so on your right here, I just have a structure of a type of protein that your cell will likely have to make at some point. And this protein here is a well-known protein that probably all of you have actually seen without knowing it. This is called hemoglobin. This protein allows the human body and the body of many animals to go ahead and take oxygen from the air and deliver this through our bloodstream to muscles and other organs that need it. And fun fact, this protein here and its cofactor called heme are actually what makes blood red. Who knew? So we've talked a lot about this idea of using cellular machinery, using cellular machinery to convert information within the cell into building blocks, as well as using cellular machinery to actually copy the genetic information of a cell, which is DNA. So let's bring this back to SARS-CoV-2. So it turns out SARS-CoV-2, which is the virus that causes the coronavirus, doesn't use DNA. Rather, its genome, or all of its set of instructions contained within the virus here that allows it to proliferate, are encoded in RNA. So the same type of species that your cell used to transmit information short distances from the nucleus to pieces of cellular machinery. What's lacking inside this virus, however, is any of that cellular machinery that normally a cell uses to replicate. And so what this means is that two things. First of all, without a host cell, SARS-CoV-2 cannot grow. So you can't have a virus in isolation in a bottle and expect it to replicate itself. It just can't. It doesn't have the ability. Secondly, this tells us that the virus actually has to hijack the cellular machinery of your cells in order to reproduce itself. That being said, though, because this virus is using RNA instead of DNA, which other forms of life use to house its genome, it'll need to replicate its information such that it can spread in a slightly different way. And because it replicates its information in a slightly different way, it offers scientists a target so that we can fight the spread of this virus. And we'll talk about what this means in a second. So in this figure here, I want to talk about how SARS-CoV-2 is able to replicate itself using the machinery of your cells. And so the way in which a coronavirus infection winds up working is that a coronavirus viral particle will enter your body through small droplets that a person who is infected is exhaling or sneezing, or when they're talking, or when they're singing. These small little droplets contain viral particles which are then able to enter your body through your lungs or mucous membranes. And then when they get to the surface of the cell, like I'm showing right here, they bind to something called a receptor. This receptor is referred to as an ACE receptor, or angiotensin converting enzyme receptor. And generally speaking, what this receptor is involved in is regulating your blood pressure. But when the coronavirus binds to this receptor, your body's like, okay, cool, you can come in. And so it tricks your body into thinking that it is actually something else, and it enters into your cell. As soon as it enters into your cell, it's going to spill all of its viral genome, which is RNA, into your cell. However, because your cell is used to seeing RNA, specifically as small copied segments of DNA, it doesn't necessarily, at this point, sort of rouse the suspicion of your cell. That being said, the virus doesn't have the ability to copy its RNA, because remember, it doesn't have cellular machinery. Unfortunately, at least for the virus, the cell doesn't have the ability to copy RNA as well. Remember, anytime your cell needs to make RNA, it's going to copy that from DNA. So what the virus actually does is it tricks the cell into making its own cellular machinery copy RNA. So for example, right here, you've got the genome of the virus, which is RNA. It is going to be translated into a protein using the cell's ribosome, which is a piece of cellular machinery. This protein is called RNA-dependent RNA polymerase. A little bit of a mouthful here. This is typically abbreviated as RDRP. What RDRP is able to do is it's able to take the viral genome here, which is made up of RNA, and it's able to essentially just start Xeroxing it. It's able to make multiple copies of both the entire viral RNA, which it will give to its replicated viruses, but it's also able to make copies of smaller portions of this RNA. So these smaller portions, which are termed subgenomic transcripts, simply because they're smaller than the entire genome, code for different proteins that the virus needs to put itself back together. Things like the capsid protein, things like the spike protein, things like the membrane and envelope proteins that actually house our viral RNA. And so after all of this is done, what the virus starts being able to do is it finds all the different parts it needs, from its structural proteins to its membrane proteins, plus its copied viral RNA. It reassembles itself into something that's called a viron, right here, number 7, and then it starts exporting this virus outside of the cell so that it can go to the next cell and start replicating itself more. And so in reality, what your cell has had happen to it here is it has been turned into a virus-making machine. And so your body is actually kind of making yourself sicker here by helping the virus replicate itself. And so a really important step in this replication cycle is this replication of RNA that is caused by this RNA-dependent RNA polymerase, or RDRP. And the reason that this is important is that this process here doesn't have any analogs in your body. What this means is your body doesn't copy RNA. And so if we as scientists could figure out a way to shut this enzyme down, to keep it from copying RNA, then we might be able to slow the virus without hurting your body. This is what makes the RDRP our target for small molecules to help fight COVID-19 and therefore keep people from getting as sick. Central to turning your cells into a virus-making machine is the copying of RNA, which is a virus-specific process. And as I mentioned in the previous slide, this starts with the virus tricking your body into making an enzyme that's capable of copying RNA. This is called RNA-dependent RNA polymerase. And so I've shown a structure of this enzyme right here. And so what this enzyme is interesting for is that this isn't used in animal cells at all. This is specific to the virus, and so this makes this a great target for scientists. So what this enzyme does is it takes a strand of RNA. So I've got an example strand here, so AAGUCG GCUUA. And so this is just the order of the nucleotides in your RNA molecule. And it's going to combine this with free nucleotides that are floating around within your cell, these building blocks of the nucleic acid. So the RNA-dependent RNA polymerase is going to copy this initial strand of RNA. So, like DNA, RNA has certain nucleotides that really like to match up with other nucleotides. A will always match up with U, while C will always match up with G. And what this means is that when we first copy our RNA, we essentially get a strand that is opposite of the strand we start with. This is called a complementary strand. What this means then is that when we copy this again, when we pair U up with A and C with G, what we wind up with is several copies of the original viral genome, because the opposite of the opposite strand winds up being the original strand. And so again, the way this works is you copy your original strand first to create a complementary strand, which is a template, and then your RNA-dependent RNA polymerase copies this again, giving you multiple copies of your viral genome. And so the interesting thing here, though, is that we can interrupt this. This is a target which we can use a small molecule to quote-unquote gum up the works of this enzyme. So the small molecule we're very interested in this talk is called Remdesivir. And so Remdesivir, which is referred to as its brand name Veklury, is a drug which targets this RNA-dependent RNA polymerase, or RDRP, of RNA-dependent viruses. This was originally developed as a treatment for Ebola and hepatitis C, and preliminary clinical data suggest that this drug can significantly shorten the hospital stay of severe COVID-19 patients, something we can all be really glad for. The way in which this works is that Remdesivir mimics the structure of the adenine RNA building block called adenosine triphosphate. And so it's this building block which is mimicked by Remdesivir that RNA-dependent RNA polymerase uses to replicate viral RNA. So let's talk a little bit more about Remdesivir. So as I mentioned in the previous slide, Remdesivir was not designed initially as a cure for COVID-19, but rather in the mid-2000s, Gilead Pharmaceuticals embarked on a project to fight a virus that causes hepatitis C. So hepatitis C is a viral infection which attacks the liver, and there are thousands of people in the United States who have this disease at any one time. And while there were treatments before the mid-2000s, these were the kind of treatments that managed the disease, did not necessarily cure it. And so what Gilead was interested in is, can we target this RNA virus, which is very similar to SARS-CoV-2, in such a way that we can cure someone of hepatitis C? And so what makes this really interesting is that hepatitis C relies on the same type of enzyme, so this RNA-dependent RNA polymerase, to copy its genome. Ultimately, there were a couple of drugs that popped out of this initial search for a cure for hepatitis C. And so the one that is the most effective, and so I've shown this with my cursor here, is called sofosbuvir. And so this is more commonly known as by the brand name Sovaldi. This is, for all intents and purposes, a cure for hepatitis C. And so you'll often see advertisements for this pharmaceutical on TV shows telling people get checked for hep C because now there is something that can cure it. And this is especially important for people to get checked simply for the case that hepatitis C is something that often manifests itself for decades in an asymptomatic fashion. So you may have also heard of Sovaldi because of a little bit of controversy that came out with the initial pricing of this drug. So one pill of a course of action for Sovaldi, so one pill originally was priced at $1,000. And so there's no real bones about it. That is incredibly expensive. One of the arguments for this was that unlike other treatments, which are maintenance treatments you have to take for your entire life, Sovaldi is able to cure a patient so that they will be well in the future. But a price like $1,000 a pill is certainly something that doesn't come without controversy. But a real big discussion of drug pricing is a topic for another day. But also popping out of this search for a cure for hepatitis C were two drug molecules, Remdesivir, as shown up in the upper left, again, brand name Veklury, and this drug fragment down here, GS441524. These showed promise in inhibiting the RNA-dependent RNA polymerase, but were not shown to be particularly effective in the treatment of HIV. So just because a certain drug doesn't necessarily work perfectly for the disease it was intended for doesn't necessarily mean that it's out of luck. And so oftentimes drugs are repurposed for new infections. And as a part of a library, medicinal chemists and pharmaceutical scientists will screen these drugs versus different targets to see, okay, can we have a drug that has worked for something else also work for something that I'm interested in? And so, again, while Remdesivir and by extension GS441524 were not effective in treating hepatitis C, it was originally then repurposed as a potential treatment for Ebola and a similar virus called the Marburg virus. And this was done as a collaboration between the United States Army Medical Research Institute and Gilead. And they found that it could block Ebola in primate studies, so think monkey studies. However, it was not as effective in treating Ebola as other treatments which were based on antibodies which were coming out at the time. So ultimately, this showed that while Remdesivir, it sort of works, it's not as effective as other treatments that you could use. But one very important thing that happened during this type of clinical trial is it provided scientists with human safety data on the drug. And so what that means is that while it was not particularly effective in the treatment of Ebola, it was not so detrimental to humans that its benefit was outweighed by its side effects. And so this human data is very important when ultimately this drug tries to get a new purpose in treating COVID-19. So again, while Remdesivir was not terribly effective in treating things like Marburg virus, hepatitis C, or Ebola, it turned out that it has a more effective action in the feline arena. So while it may not have been great at human diseases, feline diseases, well, this actually works pretty well. And so it turns out that GS441524 was investigated as a potential treatment of something called feline infectious peritonitis, or FIP, which is a terminal viral infection that's found in domestic cats. An interesting thing here is that this virus is caused by the feline coronavirus, so FCOV. So this is sort of a feline version of a coronavirus such as COVID-19. And so this research, which was done in collaboration with UC Davis and Gilead, showed that this potential drug molecule, GS441524, was extremely effective against the feline coronavirus. And so while this is certainly a different drug than Remdesivir, this is very exciting because it's shown that Remdesivir, when introduced into animal cell cultures, actually breaks down to yield GS441542. And so this is very exciting for scientists who are looking for ways to go ahead and treat COVID-19. Ultimately, Gilead has actually declined to license GS441542 for veterinary use, meaning that while it is an effective treatment for FIP, this is not something you can go out and get from your vet to treat your cat. That being said, there's a very interesting article presented in The Atlantic a couple of months ago that describes really a thriving cottage industry of labs in China who will actually make, or at least they claim, to make vials of GS441524, which they will sell to you to give to your cat. And so this article describes people using this drug to, in essence, cure their cats of FIP. That being said, there has been some controversy about this because some cats have died after taking what was purported to be this drug. That being said, there's very little quality control going on in these types of labs, and ultimately people are mailed unmarked vials for use in your cats. And so I think that might, while it might be that this drug does have side effects, I do think that it might also be related to quality control. So part of the reason why Gilead didn't license this for veterinary use, even though that seems cruel, is that because it contains the same core as Remdesivir, this meant that if there were any problems in the approval process of GS441524 towards a feline infection or a feline use, this would then have to be answered in the human trials for Remdesivir, which could complicate the use of it for treatment of humans in a pandemic. So while it's clearly unfortunate from a feline perspective, it's done such that they can ensure that this drug has fewer hurdles to get over to be used in humans. Okay, and so with the coming of the COVID pandemic, there was renewed interest in using Remdesivir as a human drug. And so preliminary data showed that Remdesivir was effective in the treatment of cell cultures treated, infected with SARS, the original virus from the early 2000s, severe acute respiratory syndrome, and MERS, Middle Eastern Respiratory Syndrome, which are both similar coronavirus infections. And so in animal models, it was very effective. And so there was also a preliminary study issued by the National Institute of Allergies and Infectious Diseases, headed by Dr. Anthony Fauci, NIAID, and it suggests strongly that Remdesivir can reduce the length of hospital stay for a COVID-19 patient. This study was, however, ended prematurely. So as a result of these early data, the FDA has issued what's referred to as an emergency use authorization, or an EUA, for the use of Remdesivir, or Veklury, as its brand name is called, for use in humans. This is not the same as FDA approval. So Remdesivir is still not FDA approved, but essentially what an emergency use authorization tells us is that given the fact that we are in extraordinary times and the benefits of this drug seem to outweigh any potential dangers, they've given this drug a pass to be used for the time being. Emergency use authorizations are not permanent, and that they can indeed be withdrawn, as was the case with hydroxychloroquine. But for now, this gives doctors the ability to treat patients really on a compassionate care basis with a drug that is believed to be the best bet that severe COVID patients have. Okay, so let's talk about how Remdesivir works. Remdesivir is referred to as what we call a prodrug. And all that means is that the initial form of the drug, as I've shown right here, this structure, is not actually the active form of the drug. Rather, it has to be converted to its active metabolite within the body. So what happens with Remdesivir is when it enters into a cell, the left part of the molecule containing this phosphate group and this ester group is cleaved off to convert Remdesivir into GS441524. And so you can see why Gilead scientists were a little gun-shy about licensing this for feline use, because it essentially is the same drug that is used in humans. And so any complications with that approval would complicate the human approval. So once you've cleaved the left side of the molecule to create GS441524, this is changed again by an enzyme in your body called a kinase. And what a kinase does is it adds phosphate groups, so POOOO groups, to your molecule to create this active metabolite that is referred to as remdesivir triphosphate. And so the reason that this drug then winds up working is that your body is going to think that remdesivir triphosphate, the active metabolite of remdesivir, is actually adenosine triphosphate, or ATP. And you can see the structure between these two molecules here and here are incredibly similar, differing by only a cyano group right here, and the positioning of some of the nitrogens on our ring, which is called a heterocycle, on the right. And so because of this, RNA-dependent RNA polymerase will actually try to incorporate remdesivir into its copies of viral RNA. So let's go back to our diagram showing how RDRP, or RNA-dependent RNA polymerase, actually replicates its DNA. So remember, the way in which this works is RDRP creates an initial complementary copy of our RNA, which the molecule is then going to, the enzyme is then going to be used as a template to create actual copies of our viral RNA. And so in the presence of remdesivir, RNA-dependent RNA polymerase tries to incorporate that molecule as a building block instead of adenosine triphosphate. And so anywhere where you see a U, RNA-dependent RNA polymerase is going to try and pair that with either an A or remdesivir. The really cool thing about remdesivir is that scientists have shown that if RNA-dependent RNA polymerase incorporates remdesivir in place of adenosine triphosphate, 3 base pairs later, so three nucleotides later, it just stops copying the RNA. Even if it's early, it'll just completely stop and then throw that viral RNA back into the cell cytoplasm. And so ultimately what this winds up causing is us to have fewer usable copies of viral RNA because we've chopped them off early. So they don't contain the entire viral genome, they just contain parts of it, which is not enough information for the virus actually to replicate itself. So with fewer usable copies of viral RNA, you're going to have fewer additional copies of new viral particles. And what this means is that you start to tamp down what we call the viral load within your patient. The lower the viral load, the less the impact that this virus can have on your patient. So this is really exciting way to help your body's own immune system fight COVID-19 by decreasing the ability of this virus to copy itself. Really cool. And this is all related to the fact that we can trick the viral enzyme RDRP into thinking that our drug, remdesivir triphosphate, so the active metabolite of our drug, is actually the building block it's looking for, but it isn't. Okay, so the reason that this winds up working so well is that remdesivir actually fits into the active site of our viral enzyme. And so Yin and co-workers showed in a very recent paper published in Science that you can actually take a picture of the RNA-dependent RNA polymerase here, and they did this using a technique called cryoelectron microscopy. This won the Nobel Prize a couple of years back, but this allows us to get a very detailed picture of the enzyme that is bound to the RNA that it's trying to copy. And so if you zoom into the center of this enzyme here, what we're able to see is we're able to see remdesivir, which is abbreviated as RMP, and it is in sort of magenta here. We can see that binding where we would expect to see adenosine bind. And so this shows from a very visual perspective how exactly this remdesivir molecule is interfering with the action of an enzyme required for COVID-19 to replicate. And so the reason that this is abbreviated RMP is because the enzyme is actually chopped off two of the phosphate groups, changing the remdesivir triphosphate into remdesivir monophosphate. So this is really an exciting time in the course of this drug, is because we can see exactly how this is binding. And the nice thing about this is that this allows scientists to ask the question, is there a way we could modify remdesivir to make it bind even stronger? Potentially. Okay, so we've talked a bit about how remdesivir as a nucleoside analog, and what I mean by that is that it mimics one of the building blocks that our RNA-dependent RNA polymerase needs to copy RNA. It mimics it in structure. So are there any other drugs out there which are trying to do this? And it turns out there is. It's a little further up the pipeline, so it'll be a little longer until this drug is available for use in humans. But like remdesivir, this drug, which is called EIDD2801, so EIDD here stands for Emory Institute of Drug Discovery out of Emory University. And so like remdesivir, this is a prodrug which mimics a ribonucleoside. And so different from remdesivir, though, is that this drug actually is present in what we call a tautomeric equilibria. And what that means is that at some times, this drug looks like our form on the top, where we've got an OH bound to a nitrogen up here, which is doubly bounded to our ring. And at some other times, it looks like a different tautomer, meaning that we have a double bond between the carbon and the nitrogen of the ring, where the carbon-nitrogen bond outside of the ring is a single bond. And so this seems like a really small difference, but what's really cool about this is that tautomer 1 mimics one ribonucleoside, whereas tautomer 2 mimics another. So in addition to tricking the body into incorporating this drug into viral RNA strands, it's able to induce mutation in the viral RNA by essentially tricking the enzyme into which base it should actually pair to. And so this is very exciting, and it appears to be very, very effective, at least in animal models. Right now, this drug is currently under phase 2 clinical trials in a partnership between Emory University and a company called Ridgeback Therapeutics. And so it's going to be a little bit while longer before we learn of the efficacy of this drug, but this is very exciting simply for the fact that unlike remdesivir, which has to be administered in an injection form, EID2801 is actually able to be taken as a pill. And so there are scientists already speculating that you could use a pill of EIDD2801, if it becomes approved, as a prophylactic treatment for people in high-risk jobs that put them in contact with patients that have COVID-19. Alternatively, if you find out through contact tracing that somebody you've been spending time with has come down with COVID-19, scientists can envision a scenario where you're given this drug prophylactically to prevent the virus from taking hold within your cells. So there's a lot of very exciting science here, which ultimately I think could really help society get past this COVID-19 pandemic. So in summary, what I hope you learned in this webinar is that viruses work differently from other forms of life. Specifically, they lack the ability to copy themselves or to make copies of themselves and to proliferate. So because of this, they have to hijack the machinery of other cells. And so in order to replicate, the SARS-CoV-2 virus, which causes COVID-19, has to copy its entire genome. So the drug remdesivir is targeted to the enzyme that is used by SARS-CoV-2 to actually copy itself. So in targeting this enzyme, the hope is that it will slow down the replication of the coronavirus such that it allows the patient to get better. So while this drug was not found to be effective for its original uses of Ebola, Marburg, or hepatitis C, remdesivir does seem to show promise against SARS-CoV-2. So by mimicking the adenosine triphosphate building blocks which are already found in your cells, the active metabolite of remdesivir is believed to inhibit the replication of the SARS-CoV-2 genome and therefore inhibit the ability of this virus to replicate itself. Thank you so much for paying attention. I hope you've enjoyed learning a little bit about the biology surrounding viruses, as well as the chemistry of the promising antiviral drug Remdesivir. If you have any questions, I would highly encourage you to attend the live question and answer session that I'll be hosting on August 6th. For more information, simply pay attention to the social media feed on North Central College. Have a wonderful day.